Hello students, today we are going to discuss some more topics of our first biology chapter, that is the fundamental units of life. In this video we are going to discuss the following topics, that is the plasma membrane or also known as cell membrane, diffusion and osmosis. Then we will discuss different types of solutions, then cell wall, cytoplasm, and at last nucleus. So let's start today's video. First of all, we will see plasma membrane or cell membrane. What is it? Plasma membrane is the boundary of each cell which separates the cytoplasm from the surroundings. That means it is these outer walls, just like the walls of our houses or the walls of a room separates the inside from the outside. The cell membrane separates the materials or substances inside the cell from its outside or external environment. So it is kind of a boundary of each cell. This cell membrane is actually present in each and every cell, and it is made up of proteins and lipids mainly. Mainly you will find that the cell membrane is made up of proteins and lipids. Also, one of the most important feature of a plasma membrane is it is selectively permeable in nature. What does it mean to be selectively permeable? Selectively permeable means it allows some substances like some minerals or gases in and out of the cell so that it doesn't lose too many nutrients or take in too many ions. It will select or particular substances mainly based on their size can enter or exit the cell, but not every mineral or every gas, or you can say not every molecule can enter or exit the cell. Only some selective or particular substances can pass through the cell membrane. That is why it is called selectively permeable, because not everything is allowed to enter or exit the cell. And why cell membrane is selectively permeable? So that our cells do not lose too many nutrients or take in too many ions, which may cause various problems in the functioning of the cell. Also, cell membrane is very flexible and can be folded, broken and reunited. Just like you must have remember, we have discussed about an amoeba. How amoeba can change its body shape. To change its body shape, it elongates pseudopodia, which is nothing but elongation of its plasma membrane and cell content in the form of arms, false arms, hence pseudopodia. So here in the case of amoeba, you can see the amoeba changes its body shape and it can elongate and fold and change its plasma membrane. So you must remember, plasma membrane is flexible in nature. Now let's see in clarity how the structure of plasma membrane actually is. So when we'll see the cell membrane or plasma membrane and magnify it, we'll see the structure is somewhat like this. It has two layers of lipids, or mainly phospholipids. Phospho because these lipids have this head which are made up of phosphate. So this is the first layer, this brinjal or purple one you see with two legs, or it is called as tails. This is one layer and this is another layer. So it is called bilayer, and in between these phospholipid layers you will find proteins like this peripheral protein because it is on one side only, and this integral protein because it is you can say it pierces through both the layers. So the proteins can be divided in such way: peripheral proteins, integral proteins. Then it may have some cholesterol in between, and these phospholipid heads, they are, you can just imagine like they are attached to each other, but they are not tightly bonded to each other, so they can move, and hence the shape can be changed, especially in the case of amoeba. So if the question is asked, what is the nature of plasma membrane? The nature of plasma membrane is it is selectively permeable. And what is, or what are a plasma membrane made up of? Plasma membrane is made up of phospholipids and proteins mainly. So this is our structure of plasma membrane. Now let's see the next topic, that is movement of substances through plasma membrane. How through plasma membrane the substances can move in and out. So mainly there are two processes that facilitate the movement of substances without any use of energy, that is diffusion and osmosis. There is a slight difference between these two. Let's discuss that. Diffusion is movement of molecules from high concentration to low concentration. Here you will see wherever the molecules are more in number, it will be called as high concentration, and where molecules are less in number, it is called as low concentration. And in the case of diffusion, both solute and solvent will move. For example, you can see this is water, and here we have red dots. Just consider these red dots as red ink. So if you will put red ink on one side, here as soon as you put the ink here, the concentration of ink is more here in comparison to here, and also the concentration of water is here more in comparison to here. That is why what will happen? Diffusion will happen, and all these ink particles or dye particles will spread out evenly. We don't have to apply any energy here. We don't have to put any energy. This takes place spontaneously on its own. And after some time, you will see that the dye completely mixes with water. And this is due to diffusion, where the particles are moving from higher concentration region to low concentration region. Next is osmosis. In the osmosis, movement of solvent only takes place. In diffusion, both solute and solvent move, but in osmosis, movement of solvent, and in biology, solvent is water itself. So movement of solvent or water across a semi-permeable membrane from high to low solvent concentration. It is also known as diffusion of solvent. The process of osmosis is also called as diffusion of solvent because here only solvent, that is water, moves from one place to another. Here you see we have some here you see this red line, dotted line. This red dotted line is a semi-permeable membrane. Semi-permeable because it allows certain things to move, and mainly water can move through it. And here we have this orangish color is water, and we have some solute dissolved here. So if this is a semi-permeable membrane, what is happening here is at first we had equal amount of water, but now the amount of solute is more in our right side as compared to left side, and vice versa. The concentration of water is more here than here. So after some time, when we'll observe it again, we will see that the movement of water will take place from its high concentration towards low concentration, and we will find this case where here the concentration of water in the solute, in here also the concentration, in simple you can say the number of particles of water and solute are in same ratio in both. So until and unless we find an equilibrium, movement of solvent will take place. So the main difference between diffusion and osmosis is here we don't have any membrane separating. But here we have a semi-permeable membrane. This is the main difference between diffusion and osmosis. Another difference is here both solute and solvent move. The solute may be gas, solid or liquid, but in the case of osmosis only the solvent or water movement takes place. So I hope you understood the process of diffusion, osmosis and the basic difference between these two. Next we will discuss types of solution on the basis of concentration. On the basis of concentration, solutions can be of three types, that is hypertonic, hypotonic and isotonic. Here hyper means more, hypo means less and iso means same. And tonic stands for strength. So this is also called tonicity. And let's discuss what happens in hypertonic, hypotonic and isotonic solutions. First is hypertonic solution. Here the concentration of solution outside the cell is less than inside the cell. That means concentration of solution means the water particles and solute particles. So the water particles inside the cell will be more in comparison to outside, and the solute particles will be more outside, and inside we will have less solute. Like you can see this picture, here we have only two solute particles inside the cell, but we have many more solid particles outside. In such cases, what happens? We know that in osmosis, the membrane of the cell will work as a semi-permeable membrane, and we know in osmosis the water movement will take place from higher concentration to lower concentration, and the concentration of water is more inside. So the water will come out from the cell and it will lead to shrinking of the cell, as cell will lose water. This is also known as plasmolysis. Then in the case of isotonic solution, equal concentration outside and inside the cell. That means the water concentration and the solid concentration will be same outside and inside the cell, and due to this cell gain equal amount of water which it lose. So as much water is coming inside, the same amount of water will exit the cell, and hence there will be no net change. So the cell will stay the same size, there will be no change. And the last one is hypotonic solution. Here concentration of solution outside the cell is more than inside the cell. That means the concentration of water is more outside than inside, and the concentration of solute is more inside than outside. Due to this, the water will start moving inside the cell more and which will lead to gain of water inside the cell, and hence cell will swell up and in some cases may even burst. So you should remember, in hypertonic solution what happens? Cell loses water and gets shrink. In isotonic solution, cell lose same amount of water as it gains and hence no change. While in hypotonic solution, cell will gain water, it will absorb water and gets bulge or swell up. So these are three types of solutions on the basis of concentration. Next we will see cell wall. Cell wall is present mainly in the plant cell and also in some bacteria and fungi, but it is absent in animal cells. The cell wall is the outermost covering of mainly the plant cell. The plant cell also have plasma membrane. Like you can see in the picture, this inner orange colored one, you can see this is plasma membrane. But along with plasma membrane, the plant cell have an additional cover or outer cover, which is outermost cover, which is called cell wall. And cell wall is a double layered structure. The outermost layer is called primary wall and the inner one is called secondary wall. The cell wall is made up of cellulose, which is a complex carbohydrate. The cell wall is rigid, strong, porous and non-living structure. It is rigid as it do not change its shape. It is very strong and it is porous. Porous means it has some pores in it through which transfer of substances can take place, and it is non-living. The cell wall is a double layer structure as we have discussed. The cell wall of two cells are joined by a layer called lamellae. You can see here in between these three cells we have this orange colored structure which is in between these three and connecting these three cells. This is called the middle lamella in singular or lamellae as in plural. So lamellae or lamella, it connect cell wall of different cells. The cell wall, you should know, is very important. Let's discuss the functions of cell wall and the process of plasmolysis. First, what are the functions of cell wall? It provides definite shape to the cell. As the cell wall is rigid and strong and do not change its shape, it keeps the shape, it maintains the shape of the cell. And as it is strong, it helps withstand changes in the surrounding medium, and it also provides strength to the cell. It is permeable and allows substances of different sizes to move across. So even though it is rigid and strong, it has pores as we have discussed in the previous slide, and hence movement of substances can take place, but it provides strength to the cell so that the cell or the plant parts do not get torn easily, and also it provides definite shape so that if it is raining heavily outside, the amount of water outside the cell may increase, and if there is no cell wall, the water will start entering the cell and the cell will swell up and may burst. On the other hand, if it is very sunny or very dry climate or weather, the water content inside the cell will be more comparison to outside, and hence if there is no cell wall, cell will keep on losing its water and it will get shrink and may change its shape. So to maintain this, we have cell wall in our plant cells. Now what is plasmolysis? When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. This phenomenon is known as plasmolysis. When the content inside the cell loses water, the cell gets shrinked. Like if you take a cotton or cotton, then dip it in water, it will absorb water, and when you squeeze it, it will lose water and get shrink. Like that, the same thing takes place in the cell itself. So this, as you can see, in the case of hypertonic solution, the cell will lose water and the shrinkage will take place. This is called plasmolysis. In the case of isotonic, no change will be there. The cell will maintain its shape and it is called flaccid. While in the case of hypertonic solution, where water enters more in the cell, it swells up and put pressures on the cell wall, hence it is called turgid. So this here water is being lost, it is plasmolysed cell. This is flaccid as the incoming and outgoing is same, and here more incoming is there, so it is turgid cell. Next topic we will discuss is cytoplasm. Cytoplasm is the jelly-like substance or fluid content inside the plasma membrane. So if this is the plasma membrane, inside the plasma membrane this whole structure is filled with fluid or jelly-like substance and it is called cytoplasm. A cytoplasm can be divided into two parts, cytosol and cell organelles. The aqueous component of cytoplasm is called cytosol. That means here whatever jelly or fluid substance we have, this are cytosol, and in the cytosol cell organelles are present. These are subcellular structures responsible for carrying out specific jobs to keep the cell alive. The cell organelles have a particular function and they also have a particular shape for them. And these cell organelles, these structures are embedded on the cytosol, and together this two is cytoplasm. So this mitochondrion, centrosome, then lysosome, vacuole, rough ER or endoplasmic reticulum, then nucleus, smooth ER, ribosome, Golgi bodies, all these are cell organelles and they are embedded on the cytosol. Together these two makes up the cytoplasm. The next topic is nucleus. Nucleus is a very important, or you can say the most important cell organelle that we have. Nucleus is also called as the headquarter of the cell. Okay, you should remember, like our brain is the headquarter or the controller of our body. The nucleus is the headquarter or controller of our cell. It is called headquarter of the cell because it directs and controls the cellular activities. Whatever is happening inside the cell is instructed or controlled by the nucleus. Nucleus is usually round in structure, and in animal cell you will mainly find it in the center of the cell, and in the case of plant cell it may be situated towards the periphery, that means towards the side. Nucleus has a double layered covering called nuclear membrane. A nucleus has two covers and it is called as nuclear membrane. Nuclear membrane is a double layered structure. Nuclear membrane has pores which regulates the movement of material in or out of cell nucleus. Like you'll see, the nuclear membrane is a double layered structure. It will be somewhat like this, and it has pores like this through which substances can enter or exit the cell. So it will have pores in between here, and through pore materials can enter or exit the cell. The main purpose of this nucleus is the nucleus contains the genetic material information for inheritance of features from parents to next generation in the form of a DNA. It is one of the most important function of a nucleus as it contains the genetic material. When an offspring is produced, you will see that the offspring shares some features with its mother and some with the father. Like you can take an example of yourself. you have some similarities with your father and some similarities with your mother and even with your grandparents. So how these features, similar features are transferred? It is through DNA, that stands for deoxyribonucleic acid. It is important, children. You should know the full form of DNA. It is, again I'll repeat, deoxyribonucleic acid. So the nucleus contains this DNA. Next we will see the genetic nucleus contains the genetic material, and the genetic material may be present in the form of chromatin, that is entangled mass of thread-like structure, or chromosome. You see, children, this is a nucleus. It has double layered structure, as you can see, double layer, and it has some pores here. Then inside you'll see there is a nucleolus, which is you can say it has some purpose in synthesis of protein. And here you can see this thread-like structure. This thread-like structure is nothing but chromatin. Children, you should remember chromatin and chromosome are actually the same thing. The main difference is their shape. When we say chromatin, it is thread-like structure, and the genetic material is present in the form of chromatin when the cell is not dividing. But when the cell undergoes division, when the cell have to divide, all this genetic material gets condensed. They get condensed and acquire the shape like this X shape. They acquire this X shape, and now it is called as chromosome. So when the cell is not dividing, you will find chromatin, and when the cell is dividing, you will find chromosome. The functional segments of DNA are called genes. DNA is a very long thread, you can say, and it has functional units. Not all the DNA are functional. It has some functional units in between, and those functional units are called genes. So for your convenience, you should remember many genes together makes up DNA, which is a long thread-like structure, and this DNA gets coiled up around a protein called histone protein. Let me write the spelling: Histone. Histone protein. The genes make up DNA, and DNA wrapped around histone protein makes up the chromosome. This is chromosome. When you'll see it, when you will unwind it, you'll see it is made up of long strands of DNA, and again DNA is made up of many genes. Here is mentioned gene 1, gene 2, like that, and each gene is made up of various, can you see these letters? These are nucleotides, and they make up our gene. So for now, you should remember our genetic material are present in the form of chromosome or chromatin, which is again DNA wrapped around the histone protein, and DNA in return made up of genes. Now, children, based on the presence of nuclear membrane in the cells can be categorized into two groups. You should remember that nucleus contains genetic material, and every cell have genetic material, but not all of them have a nuclear membrane around them. So on the basis of that, cells can be divided into prokaryotes and eukaryotes. Let's discuss some of the difference between these two. Prokaryotes are the oldest type of cell. They are the first cell, as you can say, while eukaryotes or eukaryotic cells evolved from prokaryotes. Then prokaryotes or prokaryotic cells are small and very simple in their structure, while eukaryotes are larger and more complex. They will have many organelles and each of them will be performing a specific function and many functions are going on. Prokaryotes lack nucleus in the sense they don't have a nuclear membrane, they don't have a defined nucleus. The genetic material, like here you can see this red like structure is the genetic material which is directly present or embedded in the cytoplasm. But eukaryotes contain nucleus. That means they have specific nucleus, defined nucleus which is surrounded by a nuclear membrane. Then prokaryotes usually lack organelles. They don't have cell organelles, while eukaryotes contain organelles. All of the prokaryotes are single-celled, but eukaryotes can be single-celled or multicellular. Some of the examples of prokaryotes can be bacteria, while eukaryotes can be the plants, animals, even human beings. All these are eukaryotes. But both of these prokaryotes and eukaryotes also have some similarity between them, which is both have DNA. Of course, they both have genetic material. Both have ribosomes which make up the protein. Both have cytoplasm, the jelly-like substance, and both have plasma membrane, the outermost covering. So these are the similarity between prokaryotes and eukaryotes, while these are differences between these two. I hope you understood the difference between prokaryotes and eukaryotes. So for today we will leave up to here, and in the next video we will discuss some more topics. I hope you understood all of the topics we have covered today. For now, thank you.